Okay, so hi everyone. So first, thank you to Pierre for inviting me to present some of our results in this NORA workshop. So I do this presentation in the name of a large group, as you can see in the authors. So I will present you some information and results obtained in two projects. The first one is PERLE II, funded by Fian Cole, and the second one is called ARCHE, who just started, also funded by the Fian Cole. So both concern Australians. So the first one, PERLE II, is named as selection of flat oyster families based on rusticity. In this project, we have a challenge, which was to implement a shellfish technical center in Brittany to produce Australians seeds and juveniles. In order to test them in field to try to identify different strains according to different physiological parameters. So we wanted to also to develop genomic tools to see if we could find SNPs involved in these parameters. And there's also a task, a package that we call management, because this project is linked with professional activities. So we are highly connected with the oyster farmers who help us in this project. So the main goal is to select flight oyster families on characteristic of hardiness, resilience, growth, and not only based on, for example, Bonamia resistance. So for this project, our goal was to produce biparental families. We switched from massal reproduction to biparental families. Then from these families, we wanted to set up a survey in 2 oyster beds for at least 3 years with data collection like growth, survival, parasite index, some metabolism index, in order to identify what we classically call the resistant or the susceptible families. Those families will be used to produce a second generation and we hope a third generation, but also to detect some SNPs of interest according to the physiological parameters we are interested in. And this project is also linked with another European project called Forever. Sylvie Lapeg told you about this project, to improve and restore the natural oyster beds in Brittany, with a major concern, which is the maintenance of the genetic diversity of those oyster beds. So just some picture of our hatchery. So we have a parental conditioning room where we have a lot of aquarium where we place 2 oysters and hope that we will have a female and a male to get the larvae. We have about 150 aquarium to produce those families. Then we have a specific larva room to try to get those larvae until the juvenile stages. Then we have a growing room for the juveniles, and then we transfer the oyster in our field testing sites, which are the foreshore site where we keep the oyster in oyster bags on tables, and two deep water sites when the oyster are also kept in bags and placed in cages. So just some results from what we did. We started in 2018, and so we actually have produced 4 different cohorts. The first one, where we use a lot of genitors, we got a nice survival for genitors. We have 36 spawning, but only 7 families reached the juvenile stages. We had a lot of mortality in larvae. In the second cohort, we use about a little bit more parents, but we lost a lot of them. And that's also one of the questions I will talk about later, that the genetic origin of those genitors was very different from the first cohort. And we had a lot of genitors who were dying during this production. We kept only 14 families at the end of the process. The third cohort, we got a little bit more families, 26. And the fourth, and we got also 23 families, but all of the spawning were good, and we kept all those families. So what you can see is here is the process to perform those family is better year after year. So just some results for the testing from the first cohort that we tested on the foreshore. So we know that nobody is growing flight oyster on foreshore, but we wanted to try it to see. So what you see that over the 7 families, the survival rate was 30 to 60% according to the family tested after 3 months, and go down to 2% after 20 months. But those 2% of oyster are still alive 2 years later. When we check some parameters like the length and the weight of the oyster, you can see differences between families, with some like what we call the big one, who grow very fast, and with a moderate survival. And then we see heterogeneity in the length and weight of the different families. When the result for the second cohort, among the 14 family tested, we saw a huge difference between our 2 sites, Bay of Brest with 55% of survival rate, and only 10% in the Mont Saint-Michel, which is more in the northeast of Brittany. But when we check our families, we saw a lot of variation between families, within and between sites. In terms of survival and growth, we have a lot of combination of low survival, fast growth, high survival, low growth, etc. For the third cohort, we tried the foreshore and the 2 deep sea sites, and then the results were much better, again with a better survival in the Bay of Brest compared to Mont Saint-Michel, and a very interesting survival rate for the 26 family tested on the foreshore in Brest. We didn't expect this result, and but we are very interesting by this, because it means that the oyster, even they are on the foreshore, so they can't survive, and they grow. Some of them are growing very fast. We still have the same pattern of variation between families, and we are interested by those phenotypes in a way. So for all those samples, we sample DNA for further genotyping using RADSEQ analysis, and for the cohort 1 and 2 and 3, we analyze around 1,500 individuals, a little bit more, for those genotyping to rely to the physiological parameters. So a part of this project was to develop the genomic tool. So we decided to start by sequencing the genome. So as shown by Manu and team, so we use a similar strategy, mixing long read using PacBio, linked read using the 10x technology, and shotgun. And we finally had high-C technology. And at the end, so I go very fast, you see the Scofford number evolving according with the during the assemblage strategy. And we got a final genome with the 10 chromosome very clearly, as shown by the karyotype, for a final length which a little bit things lower than what team got, 0.9 gigabase. And we made all the annotation of gene transposable element. I mean the regular work on the genome. So I won't go deep in this detail. So you can just see that the BUSCO value are very good. So we think that the genome is maybe not perfect, but at least it's very good in terms of data quality. So the strategy that we use for genotyping is not SNP array because we didn't have one. So we developed a RAD-seq approach so it's just a way to proceed. And in this RAD-seq approach, we use 4 different enzyme combinations to be able to have a better coverage of the genome at the end and generate a much more loci than if we use only one combination. So this is basically the protocol that we use. And at the end, we actually identify a lot of RAD loci, and we are working on the last stage, like the mapping of the SNAP and the study of the SNP frequency. So just to show you, we don't have a final result actually because we are still analyzing those data. We have the data for the first cohort, and we identify between 5,000 and 17,000 loci per chromosome, and for a total of about 85,000 loci per family using this RAD-seq approach. So what we are currently doing is we are mapping the SNP on the genome that we developed. And we also are developing a SNP database, which will be enriched with data issued from genome resequencing. We did some whole genome resequencing for about 40 samples at 15x coverage to develop a SNP database. And we actually are sequencing 1,500 individuals RAD-seq libraries issued from the cohort 2 and 3. We lost a lot of time last year with the COVID period, and so all the genotyping will be done in the next month for the cohort 2 and 3 to compare the samples before and after mortality. Just a slide to just about the genetic diversity. We observed a strong difference in genital survival and larvae production during the production of our cohorts. And we also had some questions related to the maintenance of local genetic diversity. So we wanted to see where the difference in terms of genetic diversity between the different genital stock that we use in our experiment. So we compare samples from Bay of Morlaix, where we have samples, a part of the genitals are coming from this bay, Bay of Quiberon in South Brittany, Bay of Brest, which is also one of the testing site. And we had a population from Cornwall in England because a lot of oysters are coming from this population and are grown by different oyster farmers in the Bay of Morlaix. So they could have an effect on the genetic diversity of the stocks that we use. And we have a out group from the population from Duxbury in Massachusetts from USA. So we performed a RAD-seq analysis, and so I thanks to Omer for the analysis. So as you can see on the Pcr analysis, the population of Brest and Quiberon are quite similar, and the population of Morlaix is very different. Even the FST value are not so high, but you can identify a specific genetic structure for the Morlaix population. So it seems that we still don't know if there is an effect of the difference in genetic diversity in our genital survival, but we decided for the next cohorts to be very careful with the origin of the genitor that we use, because if we want to then put those families in Brest or in Mont Saint-Michel, or maybe why not in Quiberon, maybe the origin, the genetic origin of the family could be a problem in the restoration of the population. So for this project, we still are producing biparental families. We have a 5th cohort in production, and we expect 2 to 3 cohorts per year until 2023. We are doing the field testing again on all cohorts, so we are following our families for many years, and we hope to implement a new foreshore site to test this hypothesis that Ostraedulis can be grown on foreshore using the same strategy than used for Gigas using bags. We will also try to first cross from our sample from the cohort 1 and 2 to get a G2 production. We identify several families which have interesting survival and growth rate. So we want to try to produce this G2. And for me, I also want to try to get much more data about transcriptomic and DNA methylation, for example, on these families linked to the physiological difference that we observe between the sites and the families. So I will try to link those SNP that we got from the genotyping with the transcriptomic regulation, but also try to see if we can see a specific response linked to the environmental parameters. And of course, bonamia is one of the parameters that we were looking for, but we think that, as we saw in the previous presentation, oyster can have bonamia, but they're stinger live. So they are like, you can be infected by bonamia and you don't die because of bonamia. But maybe in some case, environmental parameters may influence this relation. So we want to see if we can find some relation between these parameters and transcriptomic profile. So the second project we are involved in, so you take the same team and you go for a second round, is how to improve the efficiency of our biparental family production in the artery. As you can see, we have a limitation. It's very difficult to get more than 25 families at the end. So the problem that we have is that the spawning season is extended. And we observe that a lot of samples are not, they don't make gonads during this period of sexual maturation. And so the project is to improve these parameters in the artery. So to increase the number of families, we think that we have to optimize the diet of flat oyster. So we try to find new diets to improve gametogenesis. But we also want to synchronize the gametogenesis under a controlled condition. It means that we hope that if we can have our oyster with gonads almost all together at the same time, we should improve by 2 or 3 the number of families at the end. So this approach will be done by testing the effect of potential inducers of gametogenesis. And also we hope to synchronize the gamete release at the end of the maturation period. And if it works, we could reduce the period of spawning, rather than to produce 1 or 2 cohorts per year, as actually maybe we can have 2, 3, maybe 4, if we can restrict the time to produce those larvae. And as a part of this project, we also want to create an artificial restructure that we could, on which we could attach larvae directly in the hatchery and carry those reefs, so the small structure that you can carry to put them in different fields to test those families in different conditions, maybe more sites than we have, and to see if they can be used as the beginning of a restoration of reefs So we will try this in this project. So just to illustrate what we try to do to improve the reproduction is that we started to express recombinant protein from a potential inducer of gametogenesis in bivalves called calcineurin. So here is the picture of the recombinant protein that we produce. And the idea is that we made a first experiment where we injected this protein in oysters, and we checked the gametogenesis over time, every 15 days, actually. And we just, it's just the first experiment we did. We just finished it. And as a try, we wanted to test the whole process, the protein purification, injection, and see if we could see something. And the first result is that 80% of the injected oyster, they present gonads after 15 days. It's much more than in the control oyster where we inject only the protein buffer. And a very funny thing that we see that in injected oyster, you can see a shell growth. I mean, you see that they really grow, and especially after 60 days. And we don't see that in the control buffer. So we think that this protein is not necessarily a specific gametogenesis inducer, but we know that it also it's involved in different other physiological metabolism. So at least we see an effect. We actually try to quantify this effect, and it was just to try the process, but it seems to work at least. Okay, let's see. The fifth cohort that we produced, we injected these combinations of calci A and B, and we will see at the end. So in 2.5 months, we think, if it works in terms of larvae production. So what is running in this project? We just started a few months ago. So we are developing new inducer. We have a list of protein that could be a potential inducer of gametogenesis. So we want also to try different food diet and make new experiments on gametogenesis induction with a much large number of individuals to test different parameters and not only the protein. We also plan to do the transcriptomic analysis of the gametogenesis and larvae development. We already have some data from RNA-seq, especially on larvae development. Also, histological analysis gonad to try to see how those proteins affect the gametogenesis. And I'm interested by the DNA methylation profiling during gametogenesis and see if we can find some correlation or dysfunctioning in this DNA methylation that we know this process. We know that this process is involved in the regulation of many physiological traits like reproduction, but also growth, maybe why not resistance to parasite, etc. So this project is much more applied for archery to improve this production of families that we hope to increase to get at the end have a large set of families available, tested in the field that we could use for the restoration. So thank you for your attention. So I take ideas, remarks, comments, help collaboration, because we have a lot of ideas of what could be done. And in some case, we are interested by samples that could help us to understand, in our case, the adaptation to the environmental factors. Because we saw in our families that the same family in 3 different sites may behave the same, low, for example, high survival and high growth, but in some families, you have different. So we think that the environmental parameters may be also very important in this process. And it's important for the restoration of the site, because maybe we cannot think that we will have resistant families for one trait and it works everywhere. So we are looking for population living in very contrasting environment to have an idea of genetic diversity and transcriptomic profiling to see if we can have correlation between both and compare with what we have in our testing field.
Thank you, Arnaud.